I'd like to introduce the next speaker to you, Professor Denis Noble. He is an emeritus professor of cardiovascular physiology, and he is director of computational physiology both at the University of Oxford in the UK. He is a pioneer of the very important field of systems biology, and he is probably best known for the two popular books that he wrote. The first one in 2006 called The Music of Life, and the second one in 2017, just 4 years ago, Dance to the Tune of Life. And the latter formulates, in the latter he formulates the theory of biological relativity. If he's not going to explain this in his talk, then we'll ask him in the discussion. If Giuseppe laid a very sound basis for this conference, I'm afraid Denis is going to turn all our ideas of causality in life upside down. The title of his talk is 20th Century Biology Got Causation in Living Systems the Wrong Way Round. Denis, I'm very glad to have you here as a speaker. The floor and the screen are yours.
Thank you very much. Can everyone hear me? Hello? Can I be heard?
Yes, we can.
Yes, good. Okay. Thank you very much. I, first of all, was very much hoping to be present in person in Bern when I first got this invitation to take part in what seems to me to be a very important meeting indeed. I have great love for Bern because the person who inspired my work in cardiac electrophysiology and the workings of the pacemaker of the heart way back in 1958, when I started my graduate research, was Silvio Weidmann, who was precisely a professor at the Institute of Physiology in Bern, and his work was absolutely seminal. Now, very unfortunately, for family medical reasons, I can't come, and I'm delighted that I can still take part via Zoom. Yes, I do actually think that 20th century biology got causation in living systems the wrong way round, and that's going to be what I'm going to try to convince you of in this presentation. And if I can get, oh, wait a minute, we don't seem to be, my buttons aren't advancing the screen. Let me, there we go. Yes, that worked. I'll go back. There we go. Yes. A central feature of what gave rise to my conviction that 20th century biology got causation the wrong way round is that I think we cannot use association to predict causation in biology. And I'm going to give an example of that from my own work in looking at the robustness of the cardiac pacemaker. When I first constructed a model of the pacemaker of the heart in 1960, and indeed reproduced many of Silvio Weidmann's beautiful experimental recordings of electrophysiological properties of one of the pacemaker regions of the heart, I did so with about 4 ionic channels in a cell membrane. There now must be 54. What has happened since the early work in electrophysiology of the heart and its reconstruction mathematically is that we've now found there are very many more ion channels than seemed to be necessary. And that's going to be an important part of my talk. Why is it, if I could get away with making 4 ionic channels interact with the cell membrane, its voltage and ionic currents, to produce beautiful examples of rhythm in the heart, why was it necessary for evolution to end up with 54 rather than 4? Now, I think that will come a bit later in the talk. It has to do with the robustness of biological functions. And what I will show, and this is why I think association cannot predict causation, an ionic channel, which in the work I did with my colleagues about 40 years ago, contributing about 80% of functionality, meaning 80% of the ionic current responsible for generating the rhythm, would show only 15% association if you did a gene knockout or a protein block. The missing 65%, it seems to me, is a measure of the functional robustness of the regulatory network. So not only is it the case that association cannot predict causation, the difference between association levels and the functionality that we can demonstrate physiologically is itself a measure of the robustness of the regulatory network. And here is an example of that. The blue trace is the electrical voltage in this model of the sinus node. Then two of the channels, it doesn't matter what they are, one called IF, the other called IBNA, and about 80% of the electric current generating that rhythm is initially caused by that sodium channel. But as we knock it down progressively, 60, 40, finally 80, and then 100%, you'll notice that there's only a modest change in the rhythm of the heart. That's the robustness that I'm talking about. Furthermore, this kind of physiological causal analysis, as I would call it, can even reveal the mechanism by which that robustness arises. Because if I draw a line under that electrical trace, you'll notice something very interesting, which is that at the very negative potentials here, as the full knockout occurs, there is a modest degree of change in the negative direction, and it is precisely that activates the other channel to kick in. In other words, the system seems to be, one is almost tempted to say purposefully, but certainly functionally designed in some way or another by the evolutionary process to have given this degree of robustness to the cardiac pacemaker. Now, this is terribly important for our existence, because if the pacemaker was as fragile as the model I created in 1960, we wouldn't be here to be telling the story. Moreover, that kind of work has given rise to new leads to medication that can slow rhythm gently under conditions where patients need that kind of toning down, if you like, of the rhythm of the heart. Well, when I discovered this about 30 to 40 years ago in my own work, I wondered just how general this is. I call it genetic buffering because it is, in effect, the regulatory processes at a high level in the physiological system being able to buffer itself against genetic or protein knockout. And the question is, how extensive is it? Well, the answer is it's all over the place. This is a beautiful study done a few years ago by Hillenmeyer and his colleagues, looking at a much simpler organism, it's yeast, where there are about 6,000 genes, and they systematically did single knockouts through that range of genes in yeast. The result was astonishing. 80% of the knockouts are apparently silent, meaning the association score, if this was a genome-wide association study, would actually be 0. And for many of the others, it's also true that the associations would be fairly low. That gives you a feel for the general robustness of living systems. Now, the interesting thing is this: we know that many of that 80% are functional. You can demonstrate that by exposing the organism, in this case yeast, to conditions in the environment in which it lacks some essential metabolites. And what then is revealed is why those proteins and the genes that code for them are necessary. They're necessary to create the robustness. Now, we've already heard from Giuseppe's lovely talk earlier that the idea that most genes contribute to most functions and most functions depend on most genes. Actually, I found recently that even one of the founders of the modern synthesis, Julian Huxley, way back in 1942, he actually predicted that this would be the case. I'm very intrigued by the fact that somebody who was the founder of a theory of evolutionary biology that I tend to be very critical of, the modern synthesis, actually did, though, foresee this particular point. It's so clear from the genome-wide association studies that many genes are involved in any given physiological function, that this paper appeared just 4 years ago by Boyle and his colleagues, formulating not just a polygenic hypothesis, but actually the omnigenic hypothesis, which is precisely what Giuseppe was referring to in his talk. All genes, probably. But by genes here, I would also, just to answer a question during his discussion, I would also include all the non-coding or so-called non-coding DNA. Of course, we know it codes for RNAs and many other processes. But I would say that all DNA contributes in some way or another to many or even all functions. And robustness of biology and living systems is why many genes show very low association data. Now, there are two consequences of this that I think are very important. First of all, linear addition of such data is invalid. You see, what happens with the genome-wide association studies is the scores, the association levels are so low that in order to get a feel for the total genetic, if you wish to call it that, contribution to function, what they often do is to add all the associations together. Now, because they're not causation, that is invalid. There is a non-linear relationship between any particular component in a system and the functionality. But it's also invalid for the reasons of robustness. As I said earlier on, the level of association is not itself a good indication of the causation. And then, as a further consequence, I think the gene-centric views of development and evolution need completely revising. And one can explain that in a very simple diagram that Peter Cole and my colleagues presented just a decade ago. We can represent the DNA down here, the phenotype up here, and the environment here. And there's interaction, of course, both ways with all the physiological, biological networks, or if you want to call them gene regulatory networks, that's roughly what we mean. And you'll notice that we've also included the original idea that there was some, as it were, one way or single form of DNA type that would be responsible for a phenotype. That was the original idea in the period of the formulation of the neo-Darwinist theory of evolution. Well, we can rule that out. There is no such direct relationship. All interactions of DNA that might influence phenotype have to go through the biological networks. So the way to represent that is if we do a single DNA knockout, the biological network may adapt to that in the way that I described in the case of the cardiac pacemaker, so that the effect is either very small or even zero. That will give you, of course, a zero association score, even if that particular component is actually a fundamentally important component in that set of biological networks. So you may have to do 2 knockouts or even 3, who knows how many, in order eventually to get through to the phenotype. Incidentally, this also means that the concept of a gene has changed. Mendel's concept, which was the cause of the phenotype, includes all the concepts that Johansen introduced when he introduced the concept of the gene way back in 1909, but essentially it was the same concept as Mendel was implying, which is that it includes not only whatever is present in what we now know is DNA, but also in these biological networks. That was the original concept. The molecular biological definition, which is the modern concept, of course, is a very much more restricted one. That makes a big difference to the way we think about genetic causation. So I think we have to completely revise the concept of the central dogma. The central dogma is, of course, originally the idea that DNA forms RNA, which can then be used as a template to make protein. Of course, reverse transcription and the possibility of DNA and RNA being, as it were, self-replicated in one form or another, produced changes in the original formulation, which was done in 1958. This is the 1970 formulation. But I think even the 1970 formulation is woefully inadequate as a description of what is happening in biological systems. First, because we know there is causation from the functional networks, which is epigenetic controls of the nucleotides and proteins, and that influences both DNA, RNA, and proteins. Proteins fold differently depending upon the functional networks, what other components are present, whether they're in membranes or not. And of course, there's interaction from the RNAs and the proteins that form many of the components of the functional networks. It gets even worse, though. Not only do we have all of the downward causation in the controls by the functional networks of the components of the central dogma, it's also the case that we have a two-way interaction between the functional networks, the phenotype, if you like, and the environment. So I want to suggest to you that the implications for evolutionary biology are extraordinarily fundamental, because I think 4, no less, cornerstones of the modern synthesis are incorrect through a misuse of causality in biology. They include the Weismann Barrier. I'll come on to that in a moment. The central dogma of molecular biology, the all-sufficiency of natural selection, and perhaps surprisingly, also the neo-Darwinist rejection of Darwin's idea of pangenesis. So let's start with the demise of the Weismann Barrier. It was postulated by Weismann in the late 19th century to prevent soma changes, that's changes in the body, being transmitted to the germ-line. I looked very carefully when I was writing Dance to the Tune of Life at Weismann's work, and I asked myself the question, what experimental evidence did he have for that idea? He presented it as a very definite, it was one of the first dogma, it seems to me, in biology. I call it one of the dogmas. It was presented very firmly indeed in his 1883 lecture, a year after Charles Darwin himself had passed away. I asked myself the question, what experiments did he actually do, or did anybody do at that time, that would confirm that particular idea? The answer is, of course, that the only experiments that were done were the tail-cutting experiments that Weismann himself did, actually quite a long time after he formulated the idea of the barrier. The idea of the barrier was not dependent on those experiments. Furthermore, mutilation experiments do not check the whole question of what you might call the Markian forms of inheritance, that is, properties developed during the lifetime of the individual in its soma from being transmitted to the germ-line, because it's only functional processes that could be so transmitted. The idea that somehow all forms of mutilation get transmitted was never a serious possibility. After all, we don't inherit our father's accidental damage to his hip. We might inherit the tendency or otherwise to whether hips break or not, but we certainly don't inherit mutilation. Furthermore, and this is not perhaps sufficiently well appreciated, Charles Darwin totally disagreed with Weismann's idea. He proposed pangenesis with his postulate of gemmules. What he meant by gemmules is very clear in his 1868 book. It is that cells pour out tiny, well, he called them gemmules. It's a bit like supposing that cells pour out spores. This is a word that he used himself to describe his idea, which can then get picked up by the germline. We now know that extracellular vesicles, very tiny particles containing DNAs, RNAs, and transcription proteins, perform exactly what Darwin proposed. The vesicles have been shown experimentally by a large number of papers now to be transmitted through the so-called barrier to the germline. So I conclude that the barrier idea is a very good example of the misuse of causality in biology. I would argue that in biology, because of them being open systems, there are no barriers. There are functional boundaries, the skin, the cell membranes, and so on. And these are highly functional processes where what can pass and what cannot pass is actually controlled by the organism. The active causality lies with the system. So that takes me to the central dogma, and I've already hinted about why that has to be regarded as no longer, as formulated in 1970, it certainly no longer can be taken as a reason for excluding the reorganization of genomes and the control of genomes by the organism. As I see it, there's one sense in which the dogma is correct. It's a chemical fact about molecular level events. DNA forms RNA and can then act as a template for the protein sequence, but it says absolutely nothing about what causes DNA sequences to change. And ever since Barbara McClintock's work in the 1930s, 40s, and 50s, we've known that stress on an organism, she worked on corn and showed this very clearly with parts of the chromosomes moving from one area of the chromatin to another. Under stress, organisms can, as it were, throw the cards up in the air and try and see whether they can escape the severe stress by reforming what they have as DNA. Moreover, we now know not only that that happens, which is the idea of mobile genetic elements that we owe to Barbara McClintock, for which she won the Nobel Prize in 1981, we also know that organisms can hypermutate and shuffle their genomes when under different forms of stress. That's exactly what's happening when we get coronavirus invading our body. The immune system looks for and finds, if it's lucky, the correct DNA, and it changes the DNA through altering the process of the correction of DNA errors. Moreover, all of that is functional and it speeds up evolution, which means that evolution cannot be simply a blind process that is dependent only on natural selection. I'm not arguing natural selection does not occur. I'm arguing that it's slow. And there are other processes that cause the process to go very much quicker. And this, of course, is the reason, back to the same diagram I used earlier on, for which I would say that the central dogma is no longer a reasonable way of looking at causation in biology. Now I come to the all-sufficiency, or what Weismann called its Allmacht, the all-sufficiency would be the best English translation of that, of natural selection. This is actually the foundation stone of what I call the hardened view of the modern synthesis. I call it a hardened view very precisely because if you look at Julian Huxley's original book, 1942 book, Evolution, the Modern Synthesis, he actually did not think that natural selection was all sufficient. That's a very important fact. The foundation stone of the modern synthesis was not hardened in this way. Now, I'll come to why that is the case a little bit later and when it got hardened. Darwin, though, would have totally disagreed. His sexual selection idea, formulated in his 1871 book, was that there was intentional causation by organisms capable of choosing mates. Why, in his view, does the male peacock have such beautiful plumage that female peahens had chosen that to be the case? We, of course, do it as breeders because we're doing intentional selection all the time when we generate new varieties. So I would say that natural selection is not itself an active cause. It's more like a passive sieve, and it's the fallback for variation to occur by random mutation, but it's not necessarily the fastest way in which organisms can change their genomes in order to evolve. And then finally, I come to the fourth plank, which I think has now fallen, which is the rejection of Darwin's pangenesis. I'm going to take that just a bit more slowly because this is relatively new. Darwin knew, even in his 1859 book, The Origin of Species, that use and disuse, which he accepted as a process in evolution, would require causation from the soma to the germ-line. Furthermore, contrary to what you'll find in most of the evolutionary biology textbooks, he took that idea so seriously in the later stages of his life that he collaborated for many years with the physiologist George Romanes to prove his idea with hybridization experiments. I think that if Charles Darwin were alive today, he would celebrate the discovery of symbiogenesis, which is the most spectacular hybridization ever, the complete fusion of different organisms to form the eukaryotic cells that we have today. And he would also celebrate the fact that the extracellular vesicles cross the so-called Weismann barrier and influence the germ cells. He would say, surely, these are the gemmules that I supposed must exist. So I finally come to what I think we need to do about this, because if I'm right, we have the four major planks of causality in evolutionary biology, not only wrong, but completely wrong. We've got them the wrong way round. And I think what needs to happen now is that integrative physiological understanding of organisms needs to come to the rescue. And that's why with Peter Hunter, a colleague in the Physiome project in New Zealand, I wrote this little article. It's only a 3-page article in the journal Epigenomics on how to link genomics to physiology through epigenomics. And the problem is this: if the association levels are, as we now know, so low, how do we rescue the concept of causation from the genome-wide association studies? Because, as you'll find in some of the more recent literature looking at what justifies the idea that somehow or another association gives you causation, if it gives anything at all, is the conviction that it must do so somehow or another. And I don't think it can. for the reasons I explained earlier. Even an extremely low association level can be associated with a very major causal role. And what Peter Hunter and I in this article pointed out is that it's already the case that physiological network modeling, computational modeling of complex networks is ready to answer many questions to do with to what extent are organisms capable of buffering genetic change. And then you'll be able to explain why many of those genome-wide association studies are so low. We have one or two other suggestions in that article too. So I will just finish the talk by summing up what I've been saying here and what the way forward might be. First, that statistical association does not necessarily entail biological causation. Biology is therefore not just the chemical consequences of DNA sequences. Much more than the genome is involved in controlling protein function. But the reverse is also true. And I think this ought to give the genome-wide association people a little bit of encouragement, because even a negligible association score, even a 0 score, does not mean lack of a biological role, because the networks are capable of working even when a key protein or its gene are absent, because other parts of the network simply take over. And then in the article, we outline two ways in which physiological modeling could explain the difference between association and causation. The easy part is this one, just to tackle some good examples, as I showed you earlier on with the cardiac pacemaker, where physiological experiments and the modeling based on them can show important causal roles for particular proteins, even though the genome-wide association studies show low correlations between the genes involved and disturbances of function. And the prediction would be that the models may be able to show the multi-network properties that generate such robustness. We're beginning to accumulate examples of precisely this. And it's exactly that approach that was using, that approach is precisely what was used in the computations I showed you earlier on the heart's pacemaker. But now comes the future, the more difficult part, because we have a big problem with the genome-wide association data. This is the difficult part. Could the results from genomics, which is now, of course, a vast amount of data which has been accumulated in the genome-wide association data banks, could the results from genomics be used directly to inform physiological modeling? Now, the problem is this. That would require much more than the association studies, because that simply tells us what correlations there are between the presence or absence of particular DNA sequences and physiological function. Physiological modeling, which is what would enable you to work out the extent to which a network can buffer genetic change, is and requires information on the extent to which each relevant gene is expressed. In other words, we need all the proteomics data as well. So genome-wide association studies will not be sufficient on their own. We will need to have the corresponding proteomics data. These are very difficult challenges, because I think they are nevertheless necessary, because without physiological explanation, I would argue that GWAS tells us very little. It's very important that we did the genome sequencing. I'm not arguing against that. It's been of tremendous help in, for example, clarifying the trees of life and the ways in which the organisms that we know exist today were connected together through the evolutionary process. I'm not arguing against genome-wide association studies. I'm simply saying that they're far from sufficient from defining causality in biology. So that's the end of, or near the end of my talk. Just a further bit of reading. The article with Peter Hunter is the one I've already highlighted. That's just a 3-page article and can be absorbed in a couple of minutes. Then the deconstruction of the 4 major planks of the modern synthesis as it became hardened around 1970, in other words, about the same time as the formulation of a new formulation of Crick's central dogma of molecular biology, the deconstruction of the 4 major planks is in this article published this year in the journal Biosemiotics. That's also got a large number of commentary articles in the issues of Biosemiotics this year, and also my own response to that, which is what the future is for evolutionary biology. So I hope I've left plenty of time for discussion, and that's the end of my presentation, and I will stop sharing screen. Thank you very much.
Thank you so much, Dennis. One of the advantages of having a hybrid conference is that we can actually show our appreciation by applause. That was grand. I wish I'd had you as a teacher when I was at university learning molecular biology, because you give me a much better impression of being somewhere near reality than my teachers at the time did, particularly, I think, because you're showing up the uncertainties in our science. There are several questions already. I have one myself, but I'll let the others go first. The first one was in the chat to me personally from Ulrich Löning, whom we heard before, an ENSO member from Scotland. He's asking to speak up. So, Lucas, can you give Ulrich the word? And then Siguna is next.
Okay, can you hear me?
Yeah.
Yes, that was brilliant, Dennis, and it somehow sank into my mind of something I wanted to hear. You know, it was just right, and you crystallized it beautifully. Where you finished at the difficult stage of taking it further, I'd wonder whether I dare propose a suggestion of how one can approach that. And that is to use the central dogmatic people at their own experiments and do it their own way and show that it's wrong. If you take genetic engineering and you stack 2 or more properties, the finding is that the proteomic change is of the 2 together is more than and different from what each of them alone will do. So that means either that engineered genes talk to each other and affect each other, or that all genes talk to each other and affect each other. There's no way out of one of those possibilities. So I wonder whether that can be followed up. I've been wanting to for a long time, and you've put a good reason for doing it. And there are good examples. I know the one from Brazil. We've done it at both levels, both ways of doing it, with old-fashioned gene engineering by adding things, which does complicate the issue. And now with CRISPR, which complicates it less, but and looking at the transcriptome, not the proteome, so one stage more direct. And I don't understand their complicated papers, but if somebody can get at that, it would be wonderful.
I totally agree with you, and thank you for that point. Incidentally, when I referred in the lecture to people already doing the kind of work that I want to see happening, there's a very recent publication that does that in the case of hypertrophy in the heart. Looking at the complicated networks involved, they refer to something very close to what you're referring to. They refer to it as crosstalk. As it were, all the various gene products like the RNAs and proteins forming the networks together with the cell membranes and other membranes are have so much crosstalk. This is what generates the functionality and the robustness. So I think you're right. We need these kinds of experiments. And I think without that, frankly, the genome-wide association studies are just so much stamp collecting.
Well,
it's just data. And I tell you, there are papers today published by the genome-wide association people arguing, but that's all there ever can be. I mean, they've taken causality so completely out of biology that they want to argue that, well, this is all we can give you. This is the data you need. Well, excuse me for being a little bit dubious about that, but then why are all the association scores, apart from the rare monogenetic and just a few genes being involved in a disease, why are they so low? They've got to explain that, and they can't, and they don't. We can explain it, and I think we've got to. But you're quite right. It's going to be very difficult. The experimentation used, for example, to do the work I've just referred to on cardiac hypertrophy was very complex, involving a lot of work to put the network together and understand the proteomic data, and also, of course, the functional ways in which that data will change according to the circumstances. It's all context dependent. So, you know, we're faced with so much interaction, so much crosstalk, that one has to ask the question sometimes, how can we actually get to understand it? But I don't think there's an alternative. We've got to understand it. It is what life is, and we can't argue that it should be simpler.
I'd like to quote Paul Nurse on this. Sorry, can you see that?
Yes, I've seen his book, yes.
Yeah, it's a very good book, but it's about biology, which is the current study of life. It's not about what is life, which is the title.
Indeed so, yes. I would have my criticisms of Paul too. I like a lot of what Paul does, but I don't think he succeeds in that book with what his aim was.
In what we would like to see.
No, that's right.
In what he would like to see.
Exactly so. And nor did Schrödinger in 1942.
There's another issue there, and it's the long. If I can have just a second, Dietrich.
Yes, go ahead.
Okay.
But leave some time for the other questioners.
Okay, yes, I'll leave time. It's an old Oxford one when Hans Krebs was still in the lab, and I was a student there, by the way. How is it there is such strong determinism for the few enzymes of the Krebs cycle when there is much less determinism for most other enzymes? One of the early lessons from Oxford biochemistry was that most enzymes do most things, and the degree of specialization is how we discover them, but it's not what their function in the cell is. And then that, of course, was amplified by Henry Catchell and Waddington in Edinburgh. So I was in the two places that really produced those two ideas. The evolution of highly specialized enzymes, where the knockout of the enzyme really is crucial, otherwise Krebs couldn't have done his experiments. That is in conflict with what you've just said superficially. I just suspect that those enzymes are very old evolutionary ones, and they've become specialized through being hardened, in your phrase, over evolutionary very long time. I don't know whether that makes sense. I will stop there.
We leave it there. Thank you very much, Ulrich, for your comments. I have 5 further questions already. The first one was from Siguna Müller, one of our other speakers, whom you'll hear this afternoon. She's the next speaker, actually, after lunch. Lucas, can you give Siguna the word? And Siguna, please switch on your camera so we can see you.
Well, hello, everyone, and thank you so much for this amazing talk. Gosh, I feel like you have said so much already. It's been such an honor to listen to your talk. Thank you. And you really focused on the role of networks rather than the believed material determinants that we are used to. And I think one of the belief boundaries that I'm generally thinking is, you know, I am inside of my skin, so that's my boundary, and then you're inside of your skin. But then you also talk about extracellular vesicles and exosomes and all of those. So I guess my first question would relate to what you feel is the role that, if any, or of the genetic material that we get through these routes, through, say, food, through, or if there is any passing on in terms of genetic material of viral components, you know, aka viral shedding, or even, and perhaps in the context of the RNA vaccines, of the shedding of components. And then if I may ask another question, I actually wanted to address this in my talk, but I have too much material. So I would prefer to bounce the question to you, is perhaps it's even less about the material determinants, but also about the repetition forming networks. So if genes repeatedly fire, if you have a certain reactions to a certain things like a form of associative learning, in this context, if, you know, in terms of viral infection or injection, any thoughts that come to mind? Thank you so much.
Well, thank you for those comments. I think you're on to a very important question here. If I've understood you, you're asking about the forms of inheritance that, as it were, almost bypass the DNA. I mean, for example, learning in the very early stages of life in the form of how organisms treat their young is an extraordinarily powerful way in which the genome actually gets marked epigenetically by the behavior of the parents, and that gets transmitted. That's the work of Michael Meany and his colleagues, of course, showing beautifully how just stroking behavior has nothing to do with whether you're passing information down through the DNA. It actually changes the part of the hippocampus that is generating some of the molecular basis of that kind of emotional response. And of course, organisms brought up like that actually then do it to their young later on. That's precisely what Michael Meany's work showed. So I don't know, that's only really responding to the last part of your point. You had some points made a bit earlier, and I'm not sure that I now remember what they were.
Well, it was in terms of genetic material passed on between species.
Oh, yes, indeed. Oh, I absolutely agree. I mean, you know, we once thought that only the bacteria are promiscuous in exchanging DNA with each other. We now know that all organisms do it. And I mean, first of all, organisms are doing it within themselves. They're exchanging DNAs and RNAs and proteins via the vesicles between not only the soma cells to the germ cells, but between the different soma cells. You can't understand how rapid metastasis in a cancer develops without knowing about that, because we've now shown, I don't mean me personally, I mean those who are doing the exosome work have shown how they can lay down the pathways through which the metastatic process can occur. What could be more important than discovering that there's that kind of sharing of DNA and RNAs amongst, as it were, all the cells of the body, taken in this case as a population of organisms, but organisms are doing it between each other. The microbiome, after all, contains much more DNA than our own cells. But isn't the microbiome ours too? I get muddled up with the way in which one wants to partition all of these things out.
And what do you feel like, I mean, in terms of the RNA vaccines in particular, RNA vaccines, if they produce or give rise to some small RNAs, I mean, do you see any concern there of shedding or, you know, passing on between, yeah.
Well, what is a virus if it is not, as it were, an extreme form of a microvesicle?
Yeah.
I mean, I don't know where viruses came from, but I can't believe that they were there before cells, because they depend, well, certainly as viruses are today, they depend on cells to be able to replicate at all. So I just wonder, you know, what exactly is, to use Darwin's term, a gemmule, or to use the modern term, a vesicle? And the pouring out of new viruses, isn't that much the same, sharing the DNA? You know, I think it gives you a different view of the overall interconnectedness of life, doesn't it? So I think you're onto absolutely the right lines.
Thank you.
Right. Thank you very much, Siguna and Denis.
For the others, thank you.
Siguna touched upon the subject of her own talk after lunch, Corona. The next one is David G. Lucas, can you give, please, David, the word? And I have 6 further questions.
Okay, I think I'm unmuted. Fantastic talk, Denis, and I've been reading your book about the dance of life, and it's been totally stimulating, particularly from somebody coming from the social sciences. And it's about that aspect. I want to ask a quick question relating to the origins of ENSA. ENSA was set up partly to help defend scientists who didn't share the dogma of the time, and who suffered consequently. And one of the roles of ENSA is to defend them. You mentioned Barbara McClintock, and I read in your book that her discoveries were just dismissed by the biological establishment for many years, and such that she gave up working in that direction. And it was 30 years before she got the Nobel Prize. Do you think Barbara would be a posthumous candidate for the proposed Henrik Ibsen Greenberg Award for harassed early warning scientists?
Well, there is, of course, a book by Evelyn Fox Keller on the harassment of Barbara McClintock. She did a biography of Barbara McClintock. It's a very sad story. I know, of course, her successor, James Shapiro, actually worked with Barbara McClintock. He's a microbiologist at the University of Chicago and has written a book, 2011 originally, but it's just coming out again as Evolution, a View from the 21st Century. He followed up Barbara McClintock's work, and he showed that genome reorganization under stress occurs in bacteria as well as in plants. And I think it's a major discovery. Where do you find James Shapiro mentioned in Futuma and Fitzpatrick's huge volume on evolution? My goodness, they've got 600 pages in which to outline the major achievements in evolutionary biology. Is James Shapiro there? No.
Yes, yes.
I get angry about this, and it's why I wrote The Illusions of the Modern Synthesis, because it's time we called time on this set of dogmas that have been passing for science for something like, well, I would say at the moment about 50 years. I'm dating it back to about 1970. Interestingly, it didn't exist in 1942, as I hinted earlier on. Julian Huxley's book is an extraordinary read. He was much more open to many other processes. He also predicted the McClintock type work. He accepted that organisms under stress change their inherited material. Of course, he didn't know in 1942 that it was for certain DNA. But no, the original modern synthesis was a much more nuanced position. It was still, I think, miscalled neo-Darwinism. Darwin would never regard neo-Darwinism as his inheritance. And so, you know, I'm sorry to be expressing my anger in public, but I think it's time some of us said, enough is enough. Not only Barbara McClintock, Lynn Margulis also completely rubbished.
Yes, yes.
Okay,
To help channel, sorry. To help channel your...
Well, I've said enough, haven't I?
No, but to help channel your anger, as it were, or your emotion into a progressive area, would you be one of the 3 top scientists I need for the jury of this award to be given to harassed early warning scientists, some of whom might get it posthumously as well? I
not only would be delighted to do anything like that. In 2014, at a meeting organized by the Journal of Experimental Biology, which was all on epigenetics, I gave the closing talk, and it was a very simple talk. I simply said, let us remember great men and women. And I documented in a slide the list of those who had been ignored, even though they had made major contributions to biology and to medicine.
Lovely, thank you very much.
Yeah.
Thank you, David, too, especially for highlighting the reason why ENSA exists, our organization. I'm so glad to be able to work towards our goals, to protect harassed, critical, independent scientists. The next questioner is in this room. It's the previous speaker, Giuseppe Longo. Lucas, can you please turn the view on?
Thank you very much, Dennis, for your talk. I would like to go back to the early first slides you gave that recalled to us your fantastic work of many years ago on cardiac rhythms and the formation of the vascular system and heart. Embryogenesis, something which I think is absolutely beautiful, but I still do not understand it in my own language, as it is a crucial transition to a fundamental organ that causally is very complex to understand. In what I could see, there is something that happens during embryogenesis when at once there is enough microtubules, enough gene expression, enough thickness of membranes, all these things get together, and there is a causality that at all levels of interactions produces the new phenomenon, I mean, the new organ, and so on. I like to understand this as a critical transition. So there is a continuous growth of certain components, then suddenly, you know, like in the formation of a snowflake, for a given parameter, temperature in a given context, of humidity, of pressure, for a given parameter that varies continuously, you suddenly have a snowflake, which requires the understanding, mathematical understanding, by discontinuity in a derivative. It is a derivative that changes the first or the second derivative. And there is a beautiful theory there, critical transitions, where this is given by a cascade of models that approach continually, and suddenly, so the continuity of the main parameter still allows to talk of causality physically, but there is something new invented by physicists in this theory, the discontinuity of the derivative, which is very, I don't know, I would like, first, if you can, in few words, describe again, in a causal terminology, your work of the time, and also give me some more reference, because from your books for everybody, these phenomena are, you know, you present them quite quickly because it's a vulgarization, but I would love to have some more precise references.
Thank you very much, Giuseppe. I only got part of what you were saying because there seemed to be quite an echo in the transmission through the internet, but don't worry, I think I got the essence of it. So it's partly a question of what was I getting at in that modeling of the pacemaker, for example, in assigning causation in one sense, but association in another sense? Is this the issue of why association does not give us causation, or have I misunderstood?
No, you have to.
We can't hear you.
We were muted. Please start again your question.
Yes.
I would love to have a short synthesis of the causal structure that in your work explains the formation of the current system and the stabilization of a rhythm.
Right.
And then understand it, if I can, later on. I will, not here, but I would like to rephrase it in a language that I know better and see whether it fits critical transition.
I'll try, Giuseppe. I think it's a great shame I could not come to Bern. I think you and I, and others, of course, there, having face-to-face meetings and discussing over a nice glass of wine or two would be a great way of proceeding on all of this. But faute de mieux, we'll see what we can do. What I was doing, of course, with the pacemaker model and the subsequent modelings, was simply making use of something which I know was called at the time the Hodgkin cycle, which is the way in which the voltage across a cell membrane is of fundamental importance in being part of the regulatory network. People perhaps don't have a full feel for this because it's not often talked about, but the electric field across a cell membrane is tens of thousands of volts per centimeter. Under that kind of field strength, any protein will get deformed and changed. And that's, of course, how the global property, which is in this case the charge on the cell and producing the voltage field, produces the change, the gating of the ion channels. And I think that's got a better claim to causality than anything to do with what the genes are capable of doing in enabling proteins to be made to be the channels in the membranes in the first place. Now, because of the echoes occurring through the internet, I'm not totally sure that I'm answering your question, Giuseppe. So maybe you and I enter into some correspondence after this meeting, because I think the difficulty of transmission through the internet are actually partly hampering what I understood from what you were asking. But if I haven't answered the question, I've at least answered a different question, which is how on earth can it be that membranes are so powerful? They've got huge electric fields across them. They can be causes in a very, very serious sense indeed.
Thank you very much, Denis. Giuseppe looks relatively satisfied with your reply, so
okay.
Leave it at this for the moment.
I'm used to the fact that Giuseppe and I need a lot of discussion before we both become satisfied. Really
exactly. That's what this conference is about, isn't it?
Exactly.
Our apologies, by the way, for the imperfect sound. We're doing our best to improve it. The next questioner is right beside me, so I hope he'll be better to hear. Please introduce yourself. Your affiliation.
It makes me feel very comfortable to know that other people feel uncomfortable too. Okay. I want to. You had a long list about how to approach these things. And the first thing is association, and there's a very tender spot in British upper class, which is Galtonian statistics, which had the purpose to prove that general intelligence is an upper class thing. Yeah? And what happens is that, meaning or information is obviously connected to patterns. And the first thing you do in Galtonian statistics is breaking up the patterns, ripping off everything. So I'm looking at this in the project. I'm a psychiatrist. My name is Trevor Reynes, which is the same name as the founder of biology besides. But I have a site which is called Bipo Switzerland in French.ch. And there is also a revolution of arterial biomechanics or many other things. which I try to have a more tangible approach, which obviously is also connected to patterns. And I'm very interested in theories which prove that humans by nature are attracted either by one way of seeing things just in 4 dimensions, or to have an intuitive way, which is not, it's not new to you, it's Aristoteles, but you can see it also that it is commanded by mast cells, which I could have shown. So the Galtonian statistics, do you feel uncomfortable? Because there was a German, Gustav Lienert, a professor who last century discovered very clean mathematics, which is not applied to jivas, as you say. Yeah, it's absurd. It's absurd. So I think the way forward is to increase mathematical procedures. Yes.
Yes, I wonder to what extent I mean, you refer to patterns in what you say. You know, when I look at a murmuration of starlings, what on earth are they doing? I mean, I think they're conveying information. I suspect that information is, don't attack us, we're very powerful. Now, yes, exactly. I mean, it is phenomenal what they do. But does anybody yet understand the language they're using? Now, I think that there has to be some way in which we can understand better. Whales, we can exist hundreds of miles apart, yet they're talking to each other through these great communications across the oceans. What are they talking about? You know, we need to understand the patterns. I think this is what you're saying, isn't it? We need to understand the patterns that life creates, much of which appears to be just one thing after another. I mean, listen to the bird song. Birds sometimes even have to be taught to relearn their songs because they go on adding bits and pieces to them, just like musicians, Bach adding a few more bars to his music and so on. No, I don't know whether this is what you're getting at, but I think the idea that we need some way in which we can perhaps mathematically or in some other way intuit what the meaning may be of much of communication in biology. I think that's desperately needed.
Thank you very much, Dennis. I'm taking the questions in the order in which they appear to me from 3 different sides. The next one is from the room again, then Ricarda is online, then Christina in the room again, and then Hervé Lemeur.
Can we maybe put the people in the room together right after each other? That's easier for me.
Okay, my colleague is saying it's easier for him to take the people in the room together. You want them now? Okay, so Angelica is the next one then.
Diederik, sorry, I didn't get the name of the psychiatrist.
Could you say it again, please?
My name is Treviranus, which is the same name as the founder of biology. He wrote the book Philosophy of Biology, so that's the same name. It's in the DNA, yes.
Thank you. Angelica, yes.
Okay, you hear me. All right. Yeah, thank you very much. I just concur with all the praise you've heard before, Dennis, on giving that fabulous talk. And I know you and many others who are also here in the audience have been voicing similar critique of the central dogma and the offshoots, the various offshoots of it for many, many years. And yet I think we have to face the fact, and you were getting worked up about it, and so do we, that we're not penetrating this information into those circles, the applied circles, into the biotechnology circles, etc. In fact, it seems like, you know, we're even, you know, the message is not even getting stronger. And so, I mean, I keep puzzled, and we keep discussing amongst colleagues here that as well. How is it possible that such, I understand it's intriguing and intuitively very powerful message to just have a very linear explanation of things, But why is it possible to us, or we don't get our heads around it, that this is gathering still so much money and power, even though it is failing on delivering what it has promised, or is delivering very small parts of what it has promised for decades now. And this very concepts that we're critiquing is fundamentally taught still in today's universities, and it is hardwired, if you want to call it, in school education. Every biology book is starting from that. You see it everywhere. Our students today at my university, at every other university, that's what they hear. How is that possible? That is something that is blowing our minds. Other than that, there is a powerful industry that supports this, of course, but why is it not penetrating into, becoming common knowledge, that we stop teaching this nonsense and start teaching what is actually supported by the science, the best science there is?
I think my answer would be several parts to that, and I obviously share the emotional reaction to the almost complete ignoring of some extremely powerful and persuasive arguments. I would suggest two factors that may form a part of all of this. First of all, and this is what I experienced myself from the 1970s onwards, the integrative view of biology had to take a real back seat during the 1970s and onwards. The sheer prestige of the people who formulated the central dogma of molecular biology and all the results that came out from that, pouring out extremely quickly, was so great. That was one factor in the domination, if you like, by that view. The second, I think, was that they found their great popularizer, and it wasn't George Williams, who actually is the originator of the selfish gene concept. His book in 1966 is what actually got the whole idea going. It was, of course, Richard Dawkins, whose skill at writing is simply phenomenal, and skill at debating also phenomenal. But the book is a disaster. It has cemented in the gene-centric view to a degree that it really penetrates, well, we even say, into our DNA. I mean, it's incredible, isn't it? I feel that we are up against a very deep cultural problem here. It changed the face of biology so much that when Julian Huxley came in 1963, 20 years after the first edition of the Modern Synthesis, when he came in 1963 to do a revised edition, he read the work on the central dogma and he was completely convinced. And I think that is the smoking gun in the history of this. I'm in the process at the moment of writing all of this up, because I think one way to deal with the problem is to actually rummage a bit amongst the attic, if I can put it that way, of the way historically in which much of this went wrong. But there's one very particular thing that I would highlight as being the key to it all, and I outlined that in the article, The Illusions of the Modern Synthesis, in the journal Biosemiotics, which is, how did we come to think that DNA replicates like a crystal? It's not a self-replicator. The error rate in DNA replication at the early stages of the replication process in a cell is such that you will have hundreds of thousands of errors in a 3 billion base pair genome that all need correcting. How is that done? An army of mechanisms that the cell has, but the DNA does not, come in to progressively correct nearly all of those errors. So you end up with a DNA replication that is indeed extremely faithful, but it's only faithful because of what the living cell can do to a DNA of 3 billion base pairs and get it to be extremely faithfully reproduced. But this is not replication like a crystal. That was Schrödinger's original idea in his book, the 1940s book, What is Life? It doesn't replicate like a crystal. What does is the regulating mechanism in cells, which are the cell membranes and all their proteins. The lipids do exactly what he supposed. The lipids just simply add themselves to the lipid membrane. That's how lipid membranes grow. They really do self-replicate. DNA does not. And I think that if there's a central message of my article, The Illusions of the Modern Synthesis, it is that history needs to be unraveled to show where it all went wrong. Now, why did it go wrong? Nobody knew in 1970 that all of this error correction process was actually necessary. That came later, and in some, well, not exactly obscure chemical journals, but not, let's say, in Nature or Science. It was work that had been progressively put together. You know, it is so beautiful that if you ask the question, how on earth do immune systems manage to produce on call, as it were, totally new DNA sequences to be able to capture the invading virus or bacterium or whatever, it is, of course, by modifying this error correcting process. You don't actually have to create new errors. The errors are already there. You just stop them being corrected, and that's how it works. Now, when I first unraveled this by going through the history, I was just totally amazed that this has not been admitted by those who created the central dogma. Why can't they just simply admit that DNA is not a self-replicator? Therefore, the distinction, which is central to the selfish gene concept, first put out by George Williams and then by Richard Dawkins, the central concept is that the replicator and the vehicle are separate. They are not. If there's a replicator, it needs the vehicle to enable it to be accurate. And you can't have a replicator on its own. So I think what we've got to do is to say, look, the very central distinction within the selfish gene idea, the gene-centric idea, is itself deeply incorrect, and molecular biology itself has unraveled that particular error. And I think we need to state this loud and clear. I'm very deeply grateful to the journal Biosemiotics to publish what I published this year on the illusions of the modern synthesis. But what would happen if nature had done so, and why shouldn't it? That's the question. I think your organization, which I'm delighted to hear about, it needs to collaborate with many others of us who are trying to sort this kind of problem out. There is now another website that I can mention, which is the Third Way of Evolution. You can just simply type in that with no spaces between, the Third Way of Evolution. You'll find nearly 100 of us who would largely agree with what I've just presented today. This is the first time for 80 years since the formation of the modern synthesis that such an organization has existed. It is no longer possible for that group to rubbish people like Barbara McClintock, people like Lynn Margulis, Carl Woese, and I go through the list. That is important. There is now a community, and I think your organization would find great benefit from interacting with the other communities that are coming together and that could perhaps seek a way of changing this paradigm.
I have a different answer to Angelica's question.
Thank you very much, Dennis and Angelica, for the question. Thanks very much for the encouragement to answer, Dennis. For technical reasons, and with an apology to Ricarda, who's been waiting a long time, we take one more question from the room first, from Christina from Leitz.
Well, I follow up directly, and I thank you for pointing to the scandalous scientific negligence, to put it mildly, and want to contextualize it into existing power structure by telling a very short story. I happen to know young employees in the research department of Monsanto. As scientists, they were thrilled by additional unexpected proteins they found. But their directors stopped them and said, it is our strategies not to look at them. So a strategy was and is applied to obliterate the obvious. May I provoke you for a further comment? Thank you.
Well, I think no comment is what would be the best comment. What can I say to that? I mean, you see, it's been the succession of tragedies that we've observed. And just to come to the most recent one, where as the successor in many senses, certainly intellectual successor to Barbara McClintock, which is James Shapiro, the microbiology person at the University of Chicago, why is he completely ignored? I mean, this is somebody who's still alive, thank goodness. But still, look for somebody like James Shapiro in the modern textbooks of evolutionary biology. You'll find he's completely ignored. Now, I've got a theory, it's a sociological one, and I'm afraid it's funding. And I think that one of the professors at my university, when somebody thinking a bit like me gave a lecture, this particular professor was the chair of the lecture, had no answer whatsoever to the demolition of the gene-centric view that my colleague from the United States had presented. And what he said at the end of the discussion, before everybody going off to lunch, was, well, it gets us our funding at least.
Yes, God.
Isn't that the problem? It's hundreds of billions at stake. It's not trivial.
Right. Thank you very much. We'll take 3 questions from home now online. Ricarda is the first one, and then Hervé Lemeur, and then David G. Ricarda, we can't hear you yet.
Yeah, you're mute, Ricarda.
Working. Just a second.
Okay, I think now it works.
Yes, I can hear you.
Okay, great.
Good.
Thank you. That was just such a splendid lecture. It's beautiful also, the discussion, so I'm very, very grateful.
Introduce yourself, please.
Oh, I'm Ricarda Steinbrecher. I'm a molecular geneticist and biologist. I'm based here also in Oxford. I'm on the board of ENSA, and I've been working on the issue of biosafety quite a lot. So I'm in a continuous battle basically with the risk assessors, trying to bring everything down just to like the mechanics of the molecule. And I feel like what the last point came to is at the heart of it is sort of the finances, because I was going to say, like, what is it that seems to be driving it in part is the wish to make it really mechanically useful, to have a control, to have the predictability, the illusion of predictability, because one can sell it, and to basically utilize it in a way so that we can sort of engineer our life forms, our systems, and completely ignoring, like in the beginning, like how long did it take before ecology in the biological sciences was accepted as a proper field and as something real. And we still haven't got that for gene ecology, like that it's so much embedded, like between each other and as part of the system. It's also not accepted. And it will take some time. The problem is that I feel that there's so much money behind it. And now that is where my question comes in. If you look at the current, the latest version of the attempts of modifying and trying to create the species that are envisaged, is the so-called genome editing, the CRISPR use of just hacking splitting the DNA somewhere, and then exactly, the repair mechanisms come in. And then the thing is, of course, with the CRISPR, it will cut again and again and again until there is, of course, a wrong repair, and then that is what is the result. Now, the reason why now the deregulation is starting to take place is because it's being sold as this will solve our future in terms of we can create all the crops that are drought tolerant, climate resilient, disease resistant, fast growing, carbon guzzling. It is that whole concept of, if we don't use this, we will basically not be able to feed the world. And it is exactly the fact that genes do not work exactly in isolation, that so far with the previous version of genetic modification have actually shown that we haven't been able to create any of that because biology works differently. But there's so much money going into this, so we are therefore now going into complete wrong direction in our research and our the doings. How do you think one could bring across that this concept of being able to create specific traits via individual genetic modifications is just an illusion?
It's terribly important we do so, though. I was utterly shocked when I found in the 1963 second edition of Julian Huxley's work that even in 1963 he was praising the virtue of improving the human race with eugenics. Mm-hmm. What are we doing with CRISPR? Now, it's not eugenics as it was originally conceived of, which is you just don't allow those who have the wrong genes to breed, or you eliminate them completely. But it amounts, it seems to me, to much the same thing, which is the arrogance to think that we know how to design a perfect human or to design a perfect whatever it might be. And of course, with the usual promises that this will be the savior of many of the problems that we have in the world. I would say just one thing to all of that. Look at what we were promised by Francis Collins in 1999, when they got towards the stage when the first human genome sequencing project successfully announced its conclusions in 2001. I tell you, it's worth reading the 3 or 4 lines of his speech. It was an extraordinary prediction of a cure for all the major diseases of mankind. We have not seen a cure for a single disease that matters in the sense of not being one of those that can succumb, and there are very few of them, to single molecule interventions. Now, this 20 years later, how long do we have to go before governments at least start to twig that this didn't work? That we actually spent hundreds of billions not to produce what was promised in 1999, but to completely fail to produce it. Now, I took part in a debate a few years ago at Ross-on-Wye, where one of the biggest cultural events occurs every year, together with a genome scientist from Imperial College. And I was challenging precisely this kind of prediction. And the response was, well, we need to make sure we sequence all of you. Looking at the audience, there's no end. You know, it's a self-propagating process. You know, we sequence. I lectured 2 years ago before the pandemic to a big genome sequencing conference in Ulsan in Korea, where they're sequencing 10,000 Koreans to try and get better statistical significance to the very low association levels. There was utter shock at the end of my lecture. But did it change anybody's view? I doubt it. I mean, it is, I sum it up again, you see, with the comment from a colleague here in Oxford University chairing a meeting in which this was discussed. Well, at least it gets us our funding. I don't know what to do about it. I do think, though, what I said earlier on, which is that at last, there are a number of organizations, your organization, the one that I've helped to form with Jim Shapiro, the Third Way of Evolution. We are now a community, and I think that's the beginning. You cannot change a culture if you don't have a community of people who are determined to try to cooperate together to see that it happens. And I think the cooperation is why I regret not being able to come to Bern. The cooperation that works through an interaction, generating this idea, that idea, new way of doing this, new way of doing that. It's terribly necessary. Nobody can do anything like this alone. It's you know, you'll be a lone voice crying in the wilderness. So I know I'm emphasizing what I've already said, but cooperation amongst the groups that are trying to see whether we can do something better for humanity than to fail in the way we have abysmally failed with the GWAS project. I think that's the only way forward. And at last, there are communities that have come together. Mm-hmm. That's the positive sign, I think.
This is the head of NIH, not Shapiro.
Thank you very much, Dennis and Ricarda. It's nearly lunchtime. Actually, it is lunchtime already, but I'd like to take the last two questions as well. One is in the chat. It's the penultimate chat comment to everyone, so you can see it yourself if you wish, Dennis, but I'll read it out. It's from Hervé Lemeur, and it says, it is fine to try to improve the way biology thinks itself, but if it improves the way organisms are scrambled in a not so distant future, I wonder whether it is your deep motivation. Yet it could be the consequences of your work. Do you consider it to be a possibility? Do you take it upon yourself?
I didn't quite understand that question.
No, I've asked Lucas to put Hervé on screen if he can to explain the question further. He's trying to.
Yes, I suppose the bit of that question I don't fully understand is the statement, but if it improves the way organisms are scrambled in a not so distant future, can it? I don't fully understand that. Obviously, an answer to the question, do you take it upon yourself? Yes, I take full responsibility for what I'm saying, and I'm only too happy to do so. But I'm not totally sure what the issue is if it improves the way organisms are scrambled in a not so distant future. Is that the idea that we will need to do that because we're in no, I've obviously misunderstood the point.
Hervé, please explain.
Yes.
Unmute yourself, Hervé.
Yeah.
Hervé, I think there is you are unmuted within Zoom, but maybe your headphone is still not working. No.
Maybe it's your headphones.
Sorry.
Let's have the next questioner first before we try for a last time. David G was the next one, please.
Yes, okay. Am I unmuted?
Yes, I can hear you.
Okay, so just a comment on that central question from Angelica and you too, which is, why does it take so long for truths to become the mainstream, as it were? In addition to the points made, the Willful Blindness book is very good on this, as is the IPCC, the climate change group, who talk about groupthink and other kind of, sort of socio-psychological reasons why hegemony, to use Gramsci's term, another source of ideas about this, they all tend to help explain why it takes such a long time, and funding, of course, is critical too. But my question is an earlier point you made, Denis, which was to say that physiological explanations are needed to make sense of gene-wide studies, and you made that very clear, and I can understand that. But I'm worried about its misapplication in the field of public health, because when we find that A seems to cause B with sufficient strength of evidence to, as it were, take it out and reduce harm, those who don't want that to happen say, what is the mechanism of action? You've only shown that A causes B. You haven't shown us how A causes B. And as true scientists, we don't accept the correlation until we understand how it happens. Now, we know from history, John Snow makes a robust inference about cholera and water pollution, but the mechanism of action took another 35 years before it appeared scientifically. And that's the norm for public health. We know that A causes B, and yet we don't understand it for many decades. How can we stop your good idea about physiological explanations being necessary for gene-wide studies being misused?
Well, that's a very interesting question. It would be good if people wrote about it and explained the ways in which they might be misused, because obviously we need dialogues on questions like this. And yes, I think the what I call the Physiome project database, which is the many models that are now on the Physiome website that can be downloaded and used, I've no doubt that there are ways in which they could be misused. There's no doubt about that. A very early thing that I learned when teaching other people to model the heart, and there's now a huge group of people doing that. They do it much better than I can. But one of the early things that I explained to students was this, that you need to test a model to its destruction to find out what are the misuses of the model. And right. There can, of course, and you're quite right, David, be misuses of causal modeling. A very obvious one would be that if you push the limits of modeling to create a state that is way outside what could be experimentally verified, you're going to make some very silly conclusions. Yeah. And if your conclusion is, well, there's a cure for liver cancer in all of this, and it is not there, you're going to be in a mess. So I totally agree with you. I think that means two things. First of all, physiological modeling needs great, careful validation. One of the great advantages of the journal that's based on the Physiome modeling, which is simply called the Physiome Journal, which is an online journal, is that nothing can get published unless it's been through the full validation process. There's a several stage process. First, is it consistent mathematically? Is it consistent in terms of units? Is it correctly predicting the major experimental bases of the model? Is it correct in predicting further developments that it wasn't based on? All of that needs to be shown for a paper to be published in the Physiome Journal. So some of us are trying to protect the world from misuse of modeling in biology. And I think your question, therefore, is a perfectly correct one. There's no doubt that models can be misused, and mathematical analysis itself. One of the most extraordinary places where evolutionary biology modeling went AWOL was economics. Yes. Where do all the stable state modelings of various forms of economics come from. I mean, the early ones, of course, were essentially the same as the selfish gene model. You work out how many cooperators you've got, how many non-cooperators you've got, and you put it all into a mix and you solve the equations for an equilibrium state. Well, there's nothing in equilibrium. We're all far from equilibria. No, you're quite, the main thrust of your question is absolutely right. Models can be misused, so can mathematics. And we need to guard against that.
Thank you.
Thank you very much again for some great science, Denis. We're so grateful to you. I need to say two things. Giuseppe tried to intervene twice when someone else was answering. I leave it to him to make his comments in the next session because he is the moderator of the next session. And I want to stop for lunch now because we're 10 minutes behind schedule earlier. I want to urge everyone, that's my other remark before we finish now, to check Elena Roca's comment in the chat. She's extremely grateful for this session, and she's writing a book in which she will use some of the material. With that, I'd like...
About Hervé Lemaire, was it possible to get on?
You wrote a comment here, maybe you read the comment?
I've exchanged two messages with Hervé already, but there's another one from Hervé in the chat to everyone.
Exactly.
A French epistemologist did prove that immunology thought itself in a way that steered it to 40 years. Sorry, I'll leave it to everyone to read this for themselves. I'm going to take us into lunch now because there are more comments in the chat that are worth reading. Thanks for your patience. Apologies for the technical difficulties we're faced with. We're doing our best to accommodate everyone, but it doesn't always work the way we want. Thanks again. David, I'd like to invite another applause for David, please.